Bio 102, Lecture 8, students. Now we're on to embryology. Isn't that fun? This is next in the sequence. We just talked about reproduction and all the problems that were associated with reproduction. Who thought that there would be that many problems? And now we're going on to embryology to see what happens after the fertilized egg well, the egg is fertilized, and now what? And so these are a whole bunch of new things that we're going to look at. And it is called embryogenesis. Embryogenesis, genesis means to start. And embryo, of course, is a baby. Starts with the fertilization of the egg cell or the ovum by a sperm cell. So the genesis, or the beginning of creating a new individual, would only start once the other half of the DNA has been deposited. Then a whole sequence of changes occurs, which culminates in a full individual that is ready to be released into the environment. Once fertilized, the ovum is referred to as a zygote. It is not an ovum anymore. It's a fertilized cell. It's called a zygote. A zygote is a single diploid cell. Notice it's single. It's just one. The zygote undergoes mitotic divisions with no significant growth, and that this process is known as cleavage and cellular differentiation, leading to the development of a multicellular embryo. Let's look at these divisions one by one. Do remember that all cells of the body arise from the zygote, or the fertilized egg. We have about 100 trillion cells in our body. They all arose by mitosis, which is the production of two genetically identical daughter cells from one parent cell. And to go over what fertilization is, again, the definition is it's a successful fusion of gametes to form a new organism. Fertilization does a few things. It restores the diploid number of chromosomes, it determines the gender of the individual, and it initiates cleavage. Fertilization is followed by cleavage, which is a period of rapid cell division with no growth. Let's review what gametogenesis is again. So remember, we did this in the last lecture. We looked at a bunch of slides at the very end where gametes were being produced and how by meiosis the chromosome number was being halved. Gametogenesis is the production of haploid cells in the gonads where the chromosomal number is halved. Each gamete, this would be for humans, carries 23 chromosomes, one member of each pair. So you get 23 chromosomes from your mom and you 23 from your dad to get 23 pairs of chromosomes, or 46 total. Half from your mother and half from your father. Crossing over makes each chromosome different than those received from parents. And remember, when we do have gametes that form, that are haploid, there is crossing over that occurs in meiosis in both the males as well as the females. So the gametes that are formed are completely different from the parent or the person that generated them. And so therefore the zygote, which is a fertilized individual from both parents, will be completely different from both parents. And let's look at this. So this is just a review because we don't want to forget what happened in meiosis. Remember in meiosis 1, in prophase 1, the double stranded chromosomes pair up, and they're still attached at the centromeres, which you see depicted as yellow, and crossing over occurs. And then in metaphase, they just line up at the equatorial plate. In anaphase, the centromeres do not split. And in telophase and cytokinesis occurs right after that. And so then you have, this is meiosis 1, then you go into meiosis 2 where you actually divide, the centromere will split and you end up with 4 cells and each of them will be haploid. And that's depicted over here, the full sequence. So in prophase 2, you already have recombinants or chromosomes that are now mixed. There's crossing over going on. And then in metaphase 2, of course, there's lining up at the equatorial plate again. And in anaphase 2, that's when the centromere finally splits. And in telophase 2, you end up with completely different chromosomes of that individual. And then they will combine to form a zygote. So the zygote will have completely different sperm and eggs from each parent. In embryonic development, across a range of animal species, embryonic development involves common stages that occur in a set order. And this is awfully strange, but it does happen. It happens across a whole bunch of organisms. They all start out as one cell, and then they all go through these various stages which are common to all these individuals. Overall, the process of fertilization is relatively slow in mammals. The first cell division occurs about 12 to 36 hours after the sperm binds in mammals. So even though the sperm has entered and it is now fused with the nucleus, nothing happens for about a day or so. And then afterwards, a whole bunch of stuff happens. So let's look at the right side of the pane. You see embryonic development. And what you see first, there is a zygote, which is fertilized. And then there is this zygote, which is half mom and half dad. It's going to start to divide, but it won't get bigger because it hasn't actually implanted anywhere. It doesn't have a supply of food. So it's just going to divide, and to one, it will become one cell, then from one it'll become 2, then 4, then 8, and so on. And this is called cleavage, and this is after fertilization. Then all these cells start to move around, and you get a hollow ball of cells, and that's called a blastula. And then we're going to look at gastrulation, where all these cells move some more, and you get 3 layers of cells, which become the prototypes for all our cells in the body and all our tissue cells. And then finally you get organogenesis, where all the cells start to form little organs, and then you get larval stages, and then an adult. And these would be the stages we're going to look at, the ones on the right-hand side. So first of all, let's look at the egg. The egg, by itself, haploid, is generally asymmetric. It has something called an animal pole, which is going to be the future ectoderm and mesoderm, and a vegetal pole, not vegetable, vegetal pole, which is going to be the future endoderm. Remember, there are three layers that we start out with. We have an ectoderm, a mesoderm, and then an endoderm. So these 3 layers are going to form all our tissues from this one egg. The egg cell is covered with protective envelopes with different layers. The first envelope, the one in contact with the membrane of the egg, is made out of glycoproteins and is known as vitelline membrane or zona pellucida in mammals. So here is a picture. And so here what you see is this is the egg. And here is the sperm touching it, and here is the nucleus, and this is going to go into the egg and fuse up. But around the egg, what you see is the zona pellucida, which is this layer, and then a bunch of nourishing cells or follicle cells. So let's look at it one more time. So the sperm is awfully small as compared to the egg because it just has the nucleus and a tail. And all it has to do is deliver the nucleus to the egg. The egg is really big compared to the sperm because it has all the food. that is in there, which is going to allow it to develop until it finally implants, because it can develop all at once, but if it doesn't implant, it can't go any further. So in this picture at the bottom, you see many sperm trying to reach one egg, and they will, because they have no knowledge of others being there. But the minute one sperm actually makes it to the egg and penetrates the zona pellucida, then the others are simply not allowed to enter. So only one is allowed to enter. And what you see here is in this picture, the sperms, all these sperms are trying to enter, and only one is allowed to enter, and then it deposits its nucleus, and these are where the first nucleus will become. So we're going to look at what happens after that. The fertilized egg, or the zygote, starts dividing by mitosis. This is known as cleavage. This cleavage is cell division with no significant growth, and it produces a cluster of cells that is the same size as the original zygote. Okay, so it doesn't get bigger because nothing happened. There was no food to make it bigger. At least 4 initial cell divisions occur, resulting in a dense ball of at least 16 cells, and the 16-cell stage is called a morula. Cleavage partitions the cytoplasm of one large cell into many small cells called blastomeres. So you have 16 blastomeres in a morula. Here is a picture showing you that. So here is the zona pellucida over here. That's the fertilized egg, and it's going to divide into 2 and then 4, and then as you can see, ultimately it becomes a hollow ball. After cleavage, the rate of cell division slows and the normal cell cycle is restored. Very early on in Bio 101, we looked at the cell cycle and we saw that there is a phase where it grows a lot, and then at some point it decided it has to divide, and there was an M phase and the S phase. Well, the normal cell cycle is restored after cleavage. Morphogenesis, which is the process by which cells occupy their appropriate locations, involves gastrulation, which is the movement of cells from the blastula surface to the interior of the embryo, and then followed by organogenesis, which is the formation of organs. So let's continue with cleavage. The different cells derived from cleavage up to the blastula stage, which is the 7th cell division, are called blastomeres. So every cell division up till the 7th, all the cells are identical, and they're called blastomeres. Depending mostly on the amount of yolk in the cell, in the egg, the cleavage can be holoblastic, which is total, or meroblastic, which is partial. So if there's a lot of yolk in the egg, the cleavage will not occur so symmetrically, and you'll see a lot of meroblastic cleavage in animals that have a yolk sac. Holoblastic cleavage occurs in animals with not much yolk in their eggs, like humans, and mammals who receive nourishment as embryos from their mother by the placenta or by milk. In holoblastic eggs, the first cleavage always occurs along the vegetal-animal axis of the egg. So, vegetal-animal axis, this is the animal axis, yes, over here, that's the vegetal axis. So the first cleavage is always this way. And the second cleavage is perpendicular to the first. So the second cleavage is going to be this way. From here, the spatial arrangement of blastomeres can follow various patterns due to different planes of cleavage in various organisms. And I brought you a table to show you cleavage patterns of holoblastic and meroblastic eggs. So let's look at the left-hand side first, the holoblastic. Very little yolk. What you will see is radial symmetry. Sea urchins are radially symmetric, or you can see bilateral symmetry as in amphibians. Or you can see spiral symmetry as in mollusks or annelids, or you see rotational symmetry as in mammals or nematodes. If the egg is, the zygote is meroblastic, which means it has a lot of yolk in it, the symmetry is going to be discoidal, which is present in fish, birds, and reptiles, or superficial as in insects. So meroblastic cleavage occurs in animals whose eggs have more yolk like birds and reptiles. Cleavage is impeded in the vegetal pole, with the result there's a very uneven distribution of cells. They're more numerous and smaller at the animal pole of the zygote, and less numerous but bigger at the vegetal pole, because there's so much food there. It's just not easy to make cells out of it or break it apart. So going on to the morula. So the morula, the cells of the morula are at first very closely aggregated, but very soon they become arranged into an outer layer called the trophoblast, which makes the placenta ultimately, and an inner cell mass from which the embryo is developed. Fluid collects between the trophoblast and the greater part of the inner cell mass, and the morula is converted into a vesicle called a blastodermic vesicle. The inner cell mass remains in contact with the trophoblast at one pole of the ovum, which is called the embryonic pole, where the future embryo will develop. And here is a picture showing you this. So here we are. That's our zygote. And in the two-cell stage, you see 2 cells. 4-cell stage has divided into 4, and then 8, and then finally you get to a 16-celled morula. But then these 16 cells are no longer going to be a solid ball. They start moving around, and what you see is a blastocyst. And the zona pellucida starts to degenerate, but you get this outer layer of trophoblasts, which is going to become the placenta, and then an inner cell mass, which is, so that's the first division. The inner cell mass is going to be the embryo. So that's the first differentiation of cells after the morula stage. And then in the blastocyst, then you actually get to see a bunch of cells aggregating on one side, and which will form the future embryo. Right now it's just a bunch of cells. So here is a picture showing you all this, from the fertilized egg to the 2-cell, 4-cell, 8, and 16-cell stage. Then this solid ball becomes a hollow ball, which is called a blastula. And then after those cells start to have moved around, you will suddenly see invagination on one side, and that's called an early gastrula. Then you'll see more invagination and in-pocketing. So we'll look at all these stages one by one. After the 7th cleavage has produced 128 cells, the embryo is called a blastula. Now let's see, how did it become 28? 128. So from 1, you go to 2 cells, okay? Then from 2 cells, you go to 4 cells. Then from 4 cells, they divide, you get 8 cells. Oops. Then of 8 cells divide, you get 16 cells. That's the 5th division. And then 16 cells divide into 2, and there you get 32 cells. And then 32 cells divide, and you get 64 cells. And then finally 64 cells double, and you get 128 cells. So you start out with 1 cell, and the first division results in 2, then 3, then 4. This gives you 4, and this gives you 8, and this gives you 16, that gives you 32, that gives you 64, and then you finally end up with 128. All right, so that's how you would actually progress cell-wise. The divisions are here. So you have 7 cell divisions resulting in 128 cells. So now the embryo is no longer called a morula, it's called a blastula, because it's a spherical layer of cells called the blastoderm, which surrounds a fluid-filled cavity called a blastocele. So now there's fluid in there, and there's a whole cavity. The inner cell mass differentiates into a small sac called the yolk sac, and spaces appear between the remaining cells of the mass, and a cavity called the amniotic cavity is gradually developed. And this is showing you exactly that. So here you have the blastula. So you now have the trophoderm, which is going to become the placenta, and you have the inner layer of cells, but now you have a yolk cavity here, and all of a sudden now there is this other cavity called an amniotic cavity. So now you have two distinct areas. Okay, so this inner layer is the endoderm, and that is going to become the mesoderm. So let's see, this end, I know it says ento, but it really should be endo. The endoderm is going to be this layer, which will then actually grow over this and cover it all up. And then you have another layer called the mesoderm, which will come in between. And then what you're going to have is two cavities. One is going to be the yolk sac, and there's going to be an amniotic cavity. The floor of the cavity is formed by the embryonic disc, which is composed of a layer of cells called the embryonic ectoderm, which is derived from the inner cell mass and lies next to the endoderm. The embryonic disc becomes oval and then pear-shaped, the wider end being directed forward. So then, all of a sudden now, the egg is no longer going to look like an egg, and you're going to lose symmetry. So in preparation for gastrulation, which is the next stage after being a blastula, the embryo must become asymmetric along both the proximal distal axis, so the proximal distal axis and the anterior posterior axis. So it has to become asymmetric along both sides. And now it no longer will be nice and symmetrical. It will actually have various areas that look different. Near the narrow posterior end, so the caudal end or the posterior end, there's going to be an opaque streak. So over here you're going to see a sort of a streak, and that's going to be called the primitive streak, and it extends all the way about the middle of the embryo. So you will see that happening. So that primitive streak is formed at the junction between the extraembryonic tissue and the epiblast on the posterior side of the embryo and the site of ingression. At the anterior end of the streak, there is a knob-like thickening termed the primitive node. The proximal distal axis was formed when the cells of the embryo formed the trophoblast, which forms the placenta, at the proximal end, and the epiblast at the distal end. During the early stages of development, the primitive streak is the structure that will establish bilateral symmetry, determine the site of gastrulation, and initiate germ layer formation. So this primitive streak is very, very important in establishing where future things are going to happen. To form the streak, reptiles, birds, and mammals arrange mesenchymal cells along the prospective midline, establishing the first embryonic axis as well as the place where cells will ingress and migrate during gastrulation and germ layer formation. And this is a chick embryo, and it is showing you, so remember he has a lot of yolk, so it's not holoblastic, but it will still have the primitive streak. So here is the primitive streak, and what you see is a lot of activity around here. And so this is the yolk at the bottom, so this is all the yolk. And what you see is cells migrating in and going in like this. They move in. So what you have is in the this blue tissue ultimately becomes the ectoderm and things like nails and so on, whereas the endoderm is going to be this part, and that will line your internal cavities, and then we're going to have the mesoderm forming too. So all of these things happen because the primitive streak showed up. The primitive streak extends through this midline and creates the anterior posterior body axis, becoming the first symmetry breaking event in the embryo and marks the beginning of gastrulation. This process involves ingression of the mesoderm and endoderm progenitors. So progenitors are those cells that are original. They were the original pioneer cells, but they are going to get ingressed and new cells will take their place, which will become the actual mesoderm and endoderm. The ingression of the mesoderm and endoderm progenitors, as well as the migration to the ultimate position, and then these will differentiate into the 3 germ layers. Gastrulation will take place after cleavage and the formation of the blastula. Gastrulation is followed by organogenesis, when individual organs develop within the newly formed germ layers. Each germ layer gives rise to specific tissues and organs in the developing embryo. So this happens rather rapidly. So you don't have a lot of stages. You have the fertilized egg, and then you have a morella, and a blastula, and a gastrula. And once you have the gastrula, then everything is kind of done. And then you start making organs instead. Gastrulation involves the creation of the blastopore, which is an opening into the archenteron. So let's look at this again. So here we have a blastomere. A blastula, and you have blastomeres, which are all the little cells that are part of it, surrounding an empty cavity, which is a blastocele. So this is just a blastula. So this is early. And then all of a sudden, at one side, there's going to be an invagination, which will then is the beginning of gastrulation. And once you get the invagination, it is no longer a blastula, but it's a gastrula. And when this invagination deepens, then it actually looks like a little hole here, and that's called the blastopore. Okay, and this deepening pore will actually become your gut in the future. This blastocele now has gotten squashed around. So this blastopore is not an opening into the blastocele, which is a space within the blastula, but represents a new in-pocketing that pushes the existing surfaces of the blastula together. So this is the picture one more time so you can see it happening after I've talked about it and scribbled all over it. Gastrulation occurs about 16 days after fertilization. So for 2 weeks, not much is happening. You get a bunch of cells and maybe some movement of cells, but really not much is happening. The implanted blastocyst develops 3 germ layers, the endoderm, ectoderm, and mesoderm, and the genetic code of the father becomes fully involved in the development of the embryo. And then after gastrulation, twinning is impossible. So if any twins are supposed to occur, that is identical twins, that would have to be before this stage. Additionally, interspecies hybrids survive only until gastrulation and cannot develop further. So this is the idea that we saw about the barriers in reproduction, where maybe other species can mate. They might be able to mate, but if the sperm does enter the egg, at this point at gastrulation, it's just going to stop. So more development will not occur, and so you won't get an interspecies hybrid. Some common features of gastrulation across triploblastic organisms include, so triploblastic means 3 layers, yes, tri means 3. All these triploblastic organisms have these a few common features. The first one is a change in the topological structure of the embryo from a simply connected sphere-like surface to a non-simply connected torus-like surface. So torus is like a donut. Number 2, differentiation of cells into one of 3 layers. So you're going to have that. You're going to have 3 layers of endodermal, mesodermal, and ectodermal cells. And then the digestive function of a large number of endodermal cells. So endodermis usually forms your digestive system. Also, in amniotes, gastrulation occurs in the following sequence. First, the embryo becomes asymmetric. Then, the primitive streak forms. Then, cells from the epiblast at the primitive streak undergo an epithelial to mesenchymal transition and ingress at the primitive streak to form the germ layers. And the 5 basic types of cell movements that occur during gastrulation are: first, you have invagination, forming that cavity. Then you have involution, which is shrinkage. Then you have ingression, which means to come in, so all the cells start to move in. Then there's delamination, which means that the lamina or the layers of the cells start to separate out. So you get 3 layers. And then there's epiboly, which is spreading of those same cell layers. So this has to happen in gastrulation. So gastrulation is kind of important. What does EMT mean? Well, let's look at that. For cells to move from the epithelium of the epiblast through the primitive streak to form a new layer, the cells must undergo an epithelial to mesenchymal transition. So this stands for epithelial to mesenchymal transition, to lose their epithelial characteristics like cell-cell adhesion. So an epithelial cell, as you know, likes to cling to its neighbor because that's what epithelial cells do. But it has to change a little bit to become another type of layer. So these cells have to undergo some transition. Following the EMT, the cells ingress through the primitive streak and spread out to form a new layer of cells, or they will simply join existing layers. Clumps of stem cells switch off their cell-to-cell adhesion and become migratory and take up new positions within the embryo, where they again activate specific cell adhesion proteins and form new tissues and organs. So they'll turn off that adhesion, then they move, then they go glom onto somebody else, then they turn the adhesion back on so they can become a tissue. Cytoskeleton is responsible for cell migration. Gastrulation rearranges the cells of the single-layered blastula into a 3-layered trilaminar embryo called a gastrula. These 3 germ layers are known as the ectoderm, the mesoderm, and the endoderm. And this is the most important part of gastrulation. Let's look at the 3 germ layers. The ectoderm gives rise to the epidermis and to the neural crest and other tissues that will later form the nervous system. So the ectoderm is pretty darn important. It forms the brain as well as skin. The mesodermis is between the ectoderm and the endoderm and gives rise to muscles and cartilage of the ribs and vertebrae, dermis, the lining of the body cavities, the notochord, urinary system, reproductive system, blood vessels, bone, and connective tissue. So that's what the mesoderm is. It's in between. The endoderm gives rise to the epithelium of the digestive system and the respiratory system and the organs associated with the digestive system, such as the liver and the pancreas and thyroid and the parathyroid and thymus and so on. Following gastrulation, cells in the body are either organized into sheets of connected cells, as in epithelia, or as a mesh of isolated cells, such as the mesenchyme. So you can see 2 distinct types of cells at this point. And here is what you see as a blastula in its late stages. And what you're seeing right now is the amniotic cavity and the yolk sac, and you can see all these cells, and then you can see this layer of cells, and you can see another layer of cells. So you can see the ectoderm, the endoderm, and this stuff that's in between that becomes the mesoderm, this slushy looking stuff. And you can start seeing how things are moving around. So that's the primitive streak. That's on the other side. You'd have to look on the other side and of the egg. And then at this point, it probably implants. And so you would have a tail end and a front end. So here is an embryo before, so a frog embryo before gastrulation. So that's the animal pole and that's the vegetal pole, all right? And so you have the blastocele, which is the hollow area, but you have a blastopore, which is now going to organize all the cells to move around. And then once a gastrulation has occurred, you're actually going to have 3 layers of cells. So notice the red is now here, and that becomes a notochord. Notice the blue, which is here, becomes the neural plate in the epidermis. And then notice that this small area, which you see here being tucked in, is going to become ultimately the mesoderm. So these are things that come very early on, very primitive. Organogenesis. This is the process by which the ectoderm, endoderm, mesoderm develop into the internal organs of the organism. So by now, all we have are just 3 layers of cells, really nothing. Just 3 layers of cells who could do nothing. But they have to become organs before there's anything else that can happen, or an organism can be formed. So the 3 germ layers in organogenesis differ by 3 processes. They either fold, they split, or they condense. So it's folds, splits, and condensation. Vertebrate animals all differentiate from the gastrula in the same way. Developing early in chordate animals are the notochord, which induces the formation of the neural plate and ultimately the neural tube. Vertebrates also develop a neural crest that differentiates into many structures, including some bones and muscles and components of the peripheral nervous system. Morphogenesis is the biological process that causes an organism to develop its shape. So that's different from organogenesis. It is one of the 3 fundamental aspects of developmental biology, along with the control of cell growth and cellular differentiation. The process controls the organized spatial distribution of cells during the embryonic development of an organism. Morphogenesis arises because of changes in the cellular structure or how cells interact in tissues. These changes can result in tissue elongation, thinning, folding, or separation of one tissue into distinct layers. The latter case is often referred to as cell sorting. Cell sorting out consists of cells moving so to sort into clusters that maximize contact between cells of the same type. Let's look at the mechanisms of morphogenesis, which involves movement of cells. In animals, movements of parts of a cell can bring about cell shape changes or can enable a cell to migrate to a new location. The microtubules and microfilaments of the cytoskeleton are essential to these events. So inside a cell, if these start moving, that will bring about a change in the entire shape of the cell, or maybe even its migration. So if we take a look at this, so here's an ectoderm, which is going to look how the cells are thickening. Then they're invaginating, and you get a pocket, and then you finally get a neural tube. But let's look at the cytoskeleton itself. So in this picture, what you see is the microtubules are not arranged in any sort of way. All of a sudden, just before they become the neural plate, the microtubules are now arranged in a very specific way. And these are actin filaments that are going transverse, and microtubule filaments going in the longitudinal direction. And as you can see, when you're in pocketing here, they're actually moving in a way that squishes the top and broadens the bottom. And then what you see is more invagination. So some of the cells will go the other direction over here. And so these actin filaments are no longer on the outside, but they're on the inside. And you get a neural tube. So this is awfully clever and occurs naturally. So I did say that there is convergence, and I need to explain what that is. So convergence is when cells elongate and crawl between each other, as in here. So if you have a regular bunch of cells and they start crawling in between each other, that's called convergence. Extension is when the sheet of cells becomes longer and narrower. So the extension will just mean that this cell will just come and stand next to the other one, and that will go all the way inside the other one. So extension is complete movement. The cytoskeleton also directs convergent extension, movement in which a sheet of cells undergoes rearrangement to form a longer and narrower shape. Cells elongate and wedge between each other to form fewer columns of cells. Very neat. Amniotes. Land vertebrates form 4 extra embryonic membranes: the chorion, the allantoid, the amnion, and the yolk sac. This protects the embryo from desiccation and allows reproduction on dry land. So any land animal will have to do one of these things. Has to produce 4 extra embryonic membranes for the protection of the embryo, because it's no longer in water. Reproduction outside of aqueous environments required development of shelled eggs of birds and reptiles and uterus of marsupial and eutherian mammals. In both adaptations, embryos are surrounded by a fluid in a sac called the amnion. And mammals and reptiles, including birds, are called amniotes because they have these 4 layers. So let's look at this. They're all colored differently. So the chorion is purple, and that is a layer that protects the entire egg and all its membranes. Underneath that, there is the allantois, which is in green, and then there's a yolk sac, which is in orange, and then there is the amnion itself, which protects the actual embryo. So these are all protective layers. The 4 extra embryonic membranes that form around the embryo, what do? They do specific things. The chorion functions in gas exchange. The amnion encloses the amniotic fluid, which protects from bumps and as movement occurs. The yolk sac encloses the yolk, which is food, and the allantois disposes of waste products and contributes to gas change. So they all have very specific purposes. During development, individual cells, sets of cells, or whole tissues stop developing, and they have got to go. They're not needed anymore. So they are engulfed by neighboring cells, and so that's just part of it. Some they're made and then they're just eaten up because we don't need those anymore. Many more neurons are produced in the developing embryos than will be needed, so extra neurons are removed by apoptosis, which, as you recall, is cell suicide. So the cells are programmed to just off themselves. Fate mapping. This is cool. Fate maps are diagrams that show organs and other structures that arise from each region of an embryo. Cell lineage in germ layers is traceable to blastula cells. So these maps are awesome. So here you see a blastula over here, and it's color-coded, and you can see that the central nervous system is going to develop from the green area. The epidermis will develop from the blue area. Mesoderm from the red area, and you see the blastula develop into a different embryo later on, past the gastrulate stage even. And what you see is if you have a 64 cells, so this is before even the 120, the 7th division. So this is the 6th division. What you have is you can find out what is going to be the fate, because you can just put dye in one cell and then trace it, see where those cells go, and you can find out that the lineage of the cells, what do they form ultimately, because you can trace the dye. Axis formation. A body plan with bilateral symmetry is found across a range of animals. This body plan exhibits asymmetry across the dorsal ventral and anterior posterior axes. The right-left axis is largely symmetrical. The animal-vegetal asymmetry indicates where the anterior posterior axis forms, and the dorsal ventral axis is not determined until fertilization. Upon fusion of the egg and sperm, the egg surface rotates with respect to the inner cytoplasm. This cortical rotation brings molecules from one part of the vegetal cortex to interact with molecules in the inner cytoplasm of the animal hemisphere, and this leads to expression of dorsal and ventral specific gene expression. Isn't that cool? So what you see here is the three axes of the fully developed embryo on the top. You see the dorsal, which is back, the ventral, which is tummy, posterior, which is back, and anterior is front, and then you see the right and left. So those are the three axes that we're looking at. When we are establishing these axes in an egg, you don't really have that. You have an animal pole and you have a vegetal pole. And then you actually have the point of entry where the sperm entered, and that's it. What you will see is that the egg actually rotates a little bit so that the yolk cells start to move and exchange material with the animal cell. The yolk part of that cell will go and move around, so you will have some differentiation already occurring just before the first cleavage. Gravity is involved in establishing the anterior-posterior axis, and later pH differences between the two sides of the blastoderm establish the dorsal-ventral axis. So it's not magic. It's either physics or it's chemistry. So these terms are important. In mammals, embryonic cells remain totipotent until the 8-cell stage, much longer than other organisms. So totipotent is a cell that can become any other cell. But then after the 8-cell stage, progressive restriction of developmental potential occurs, and that's a general feature of development in all animals. In general, tissue-specific fates of cells are fixed by the late gastrula stage. So even if it's, not the 8-cell stage, definitely by gastrula, the cell's fates are sealed, and they don't become something else. So they're already fated to be a certain. Cell fate determination. As embryonic cells acquire distinct fates, they influence each other's by induction. The dorsal lip functions as an organizer of the embryo body plan, inducing changes in the surrounding tissues to form the notochord, the neural tube, and so on. Inductive signals that control pattern formation are called positional information. It tells a cell where it is with respect to the body axes. So first the axes are established, and then signals will tell the cells where are you with regard to those axes and where to go. Kind of clever, no? So here we are. We're going to summarize all this. The ectoderm, which is the outer layer of the cell, what does it do? What does it become? It becomes the epidermis of the cell and its derivatives, including sweat glands and hair. Ectoderm becomes the nervous system and sensory systems. It becomes the pituitary gland, the adrenal medulla, and the ectoderm becomes your jaws and teeth, and it becomes germ cells. Mesoderm, which is the middle layer of the embryo, will end up being the muscles. So it'll be the skeletal and muscular systems, the circulatory and lymphatic systems, the excretory and reproductive systems, except germ cells. And the dermis of the cell, which is the layer underneath the epidermis, and the adrenal cortex. So not the adrenal medulla, but the adrenal cortex. This is kind of cool. And then the endoderm, which is the inner layer of the embryo, will form the future epithelial lining of the digestive tract and the associated organs like the liver and pancreas, and the epithelial lining of the respiratory, excretory, and reproductive tracts and ducts. And the endoderm will form the thymus, the thyroid, and the parathyroid glands. So the endoderm doesn't do a whole lot, but it does do very key things. Here we are, we're looking at a picture. So now we're going to just look at a whole sequence of images, and I'll just narrate. So here, these are all summary slides. So we have an egg, it is going to be fertilized, and then what you have is a zygote. And then the zygote divides, and there's cleavage going on, 2 cells, 4 cells, 8 cells. Finally, you get to the morula. The morula becomes the blastocyst, and you get a hollow cavity and a bunch of cells stuck on one side. Well, at this point, um, the egg is, so also I should have mentioned that the egg is traveling down the uterus, and it is still traveling down the uterus during the morella stage, and it is still traveling down the uterus in the blastocyst stage. It is only near the gastrula stage will it will actually, on its way out, on its exit, will it attach to the uterus and become a gastrula. And so until it's attached, you can't develop any further. So attachment is kind of key. And this is showing you in sequence in days what is happening. So in about a week after fertilization, the fertilized zygote is now implanted. And once that is implanted, a week or so later, a few days later, many things will happen. And you can see that the embryo itself is now incorporated into the uterus, the lining of the uterus, and now it is going to be developed inside. Here we see week 3 and week 4 and the late week 4. So in week 3, what you're seeing is the amniotic cavity, the amnion, and the embryo. Still not distinguishable at all. All you see is a bunch of cells, and you see the primitive streak and things like that. And in early week 4, you're beginning to see this part, which is curling around to become a proto-embryo. And at the end of week 4, you actually see tiny organs being formed, the spinal cord, and so on, actually being formed. So still, it is now attached, and it's getting food, and that's why it can actually develop more. Early week 3, let's take another look at it. The primitive streak. Remember the primitive streak, very important for directing where the cells have to go. And the cells migrate, and you get this middle pink layer, which is going to become the mesoderm. So this is important to remember. And what we have here is in week 3, we have a cephalocaudal folding. So you have this folding over here, which is ultimately going to become the head, and then some folding over here, which is going to become the tail. So this is still kind of very, very fluid and not well defined at all. This is shown in the transverse section. So whereas the transverse section, you'll see the folding like this. Not much actually can be seen, but still the same rudiments are there. And ultimately, this person that was this blue curled worm-like thing becomes a full-fledged person. So here are some other views of the amniotic, the very early stages of the cells. So this is the 3rd week, the middle of that. So it's still a very, very early stages where cells are moving around, and what you see is the loss of symmetry and the primitive streak and so on, but what you see is a very strange looking egg. And if you looked very carefully, you could see neural folds forming to form the neural tube, which is currently there's a big debate over the Zika virus, and that's where the neural tube doesn't actually close up. So that is where the problem occurs, and then it never gets fixed. And the mesoderm is covered, is this layer, which is here, yes, and that is what becomes the muscles. And then the endoderm ultimately becomes all the stuff in pink that we see. And I hope that was helpful, and I'll see you next time, students.